Hey, welcome back. So, what have we covered so far? All the way back in the Greek philosophers, Plato and some of his compatriots were identifying that it seemed rational that you could go from something that was really complex and take it down to a smaller, very basic elemental level. That element at that time was nicknamed the atom. That would go on to be the foundation for a lot of our studies in physics, Newtonian physics, and our understandings of how the world operated. And we thought that it behaved in a very electric fashion, things being either positive or negatively charged, and based on how those things operated at a magnetic level, a polar level, was how stable it was. So, example, your desk, your table, piece of wood, this is a very stable carbon-based structure. It is technically organic because it does have carbon in it. But the movement of the atoms are so slow that they appear to be stable and solid. The faster the atoms move, the less likely we are to actually be able to see them. As an example, when you're looking through a microscope, you will observe kind of a slight jittery moving of things that are on the slide. That's actually called Brownian motion. It's the impact of light interacting with a liquid medium going through a magnification spectrum. So as you magnify it, it actually shows and looks like motion. The other thing that we learned is that what we thought were the smallest elements, meaning the basic atom, the electron, the neutron, and the proton, are actually also made up of smaller and smaller things. These smaller things, sometimes identified as quarks, tachyons, neutrinos, muons, taus, bosons, these we thought would behave in a very simple manner. And hopefully, if you've been able to watch the string theory video yet, you'll understand that no, in fact, these are actually quite complex. There's nothing basic or simple about any of them. And the fact that they are all still very well ordered, behave by very defined rules of operation, shows us that there's an incredible amount of structure to our universe at both a subatomic level as well as a macroscopic level. That no matter where we look in the universe, whether it is in the smallest aspects of who we are and how we are made up, it is incredibly complex and it is so coordinated, so defined, so well structured that it appears to be designed. This is why most people, if they successfully complete any level of science, they will come to understand what Louis Pasteur used to say, which was that a little bit of science would lead a person away from God, but a in-depth understanding of science would reveal the miracles of God. Just understanding the in-depth interactive manner in which things operate in the world of physics and chemistry alone would lead one to believe that things are designed. And as such, it's not a big leap to go from something being designed to having a designer. Sorry. Now, moving forward, at the chemical and elemental level, you have the elements. So we're dealing with just the atomic level right now, such as helium, hydrogen, copper, gold. Those things, they are all defined by their mass, by the number of protons, by the number of neutrons, and by the number of electrons. The number of electrons in the outer shell or valence of that electron, of that structure itself, tell us how stable or volatile that particular element is. So, sodium, even though it is a metal, is highly reactive with certain other substances to include water. If I interact the two together, it is explosive. But if I bond it with a chlorine gas, then it becomes incredibly stable and is in fact a manner in which we maintain homeostasis balance within the human body or any other living organism. When we look at three specific electrolytes, particularly charged specific atoms, potassium, the big K there, sodium, Na, chlorine, chloride, the negatively charged particle. These help us maintain how things move and communicate within the confines of our body, between the nerves and the muscle cells. Now, a covalent bond, again, we're dealing with carbon here specifically, is where not only are we going to potentially give up an electron, ionic bond, but we're going to share that electron and it holds them together. This base structure of carbon and hydrogen is something you'll see a lot, and it'll make a lot more sense later. But this is one of the strongest structures you can have when you have a bunch of these together. Now, those covalent bonds in the process of sharing between the atoms, a one covalent bond equals one pair of shared electrons. Two covalent bonds, you have a double bond, meaning they're sharing two electrons. For a single, four electrons for a double, six electrons for a triple bond. This is what it might look like at an atomic structure level. I apologize for all the little pings you may hear. People tend to text me on a regular basis, and I can't turn off those notifications. Hydrogen and water have a very strong covalent bond. And one of the reasons why it is so effective as it is called the universal solvent as opposed to a solute. A solvent breaks down other things. A solute is something that's broken down inside of something. So, as an example, water is utilized to break down other things when you're washing your laundry, you're washing the dishes, or whatever else. It can actually break apart other chemical bonds and help move things along. It is one of the strongest types of individual chemicals in the world, but it is also perfectly pH balanced. And that'll make a little bit more sense in just a moment. Polar versus non-polar covalent bonds. Again, we're sharing something. However, the outcome becomes polar in defining whether or not we have a positively charged or negatively or neutrally charged molecule at the end of the day. So the polar molecules, they're unequally shared electrons between the atoms, so they have a slight positive charge. If it's at one end, and if it's at the other end, it will be a negative charge. You have non-polar molecules where they're sharing, but it's so perfectly balanced, it's stable. Um, Water is probably the most common covalent compound, and the lines between the O and the H's indicate a covalent bond. So the way we write this single line is a single covalent bond. If I had a second line, we would be defining the double bond. And in that rare organic structure, we may see a third bond or third line written to show that it is a triple bond. Now, hopefully that all stuck. There'll be another video to help dive a little bit deeper into these things. We're going to talk about some reactions next. Oxidation, as an example, you may have heard of oxidation in other things, such as when stuff oxidizes. One of the most common things that we see that oxidize is metal. When metal oxidizes, it creates rust. So this is something where we're losing or breaking down a part of a chemical molecular structure. It's becoming something new, but what's happening is we are losing electrons one molecule at a time, and we're the reduction, the gain of electrons by a different molecule or ion. So we are losing or breaking down that chemical reaction utilizing oxygen, hence the oxidation. Now, one of the things where This plays out a huge part at the cellular level within the human body is in the confines of our red blood cells or RBCs. At the center of a red blood cell is a molecule known as the heme molecule, Heme. Within the heme molecule, it is known as a quaternary structure, meaning it is more than a four-folded chemical structure. And at the center of it is iron, the symbol of which is Fe. Now, there is a process that is going on all the time with that iron that is breaking down the iron and basically rusting it from the inside. Part of the reason why red blood cells look red is technically they're rusting through an oxidation process. Now, when you have more oxygen, it looks more red. When you have less oxygen, it looks less red. When you look at the veins on your arm or your hand, they may appear to be blue. That's simply a reaction between the light, the skin tissue, and the fact that there is less oxygen in that blood vessel when compared to an artery that is hyperoxygenated, meaning it has a lot of oxygen in it. Now, each one of these pieces is vital for you to understand, at least at a theoretical level. You don't have to have this mastered. You just need to know that it happens. So part of what we're going to be spending time on is just understanding that this happens, and we're going to revisit these elements a piece at a time when we're dealing with the different cell structures that make up the different parts of our body. So as an example, we're going to be talking about the skeletal structure next. And a part of that, a huge part of that, is that in the bone itself, the part you see, the average person sees, is that hard outer white surface. Now, technically, every time you brush your teeth, you're brushing your skeleton. It's the only time you see your skeleton outside of the body, outside of the tissue. Inside of that bone is a soft, squishy bit that is called the marrow, Marrow. So if somebody says, "Oh, I made a bone marrow broth" or something of that nature, what they're basically saying is they boiled the soft, squishy bit inside of a bone and they turned it into a soup. That soft, squishy bit is what actually produces all of the red and white blood cells that will go through your entire body. In addition, in order to maintain the structure of the bone itself, it's producing its own bone cells that are sometimes called osteoblasts and osteoclasts. Osteo meaning bone. If you understand that calcium is at the base of the cell structure required in order to build healthy bone, then you can understand that it's a result of a very strong atomic structure that bone is held together so tightly and so solidly that you're actually able to walk around and that bone will hold you upright. It is what keeps you and maintains you in as solid of a level as possible. Now, when we talk about other things, those bonds can be broken down at a chemical level and broken down into new substances. We see this a lot through our body process known as metabolism. We take one thing in, it's broken down, turned into other things that the body needs in order to produce energy, produce new parts, remove old parts, and develop even new tissues. Now, the things that cause those reactions and cause those breakdowns are going to be things like reactants that are starting materials, and the other term is a product, which is the final material. So, as an example, if I consume a big teaspoon of table sugar. It is sucrose. The chemical name, anything ending in OSE is a sugar. Anything ending in ASE is a enzyme. Just keep that in the back of your head. So sucrose is table sugar. It breaks down into two smaller sugar types, one being glucose and the one being fructose. I can't necessarily use the fructose, so it will most likely become a waste product. I can and I need the glucose, and that will be utilized to run all kinds of things in the body that we'll cover a little bit later on when we discuss macronutrients. Now, inside of the body, two very common reaction types are dehydration and hydrolysis. They sound just like what you think they mean. Dehydration means I removed water and I created a new molecule as a result of it. Hydrolysis means I added water and it created a new set of molecules. So in a dehydration synthesis, I've removed water, H2O, and I've separated that out as a separate thing. And then I have the final molecule that's aside from that water. When I deal with hydrolysis, I'm going to add water in order to produce two other smaller molecules. Now, again, basic reminder, ionic bonding versus covalent bonding is whether or not I gave up an electron or I'm sharing electrons. Hopefully that makes sense. Again, there'll be another supporting video to help you hopefully make a little bit more sense out of this. This will be coming from, I utilize a lot of Bozeman science because they're very helpful videos. And sometimes hearing a different voice explain things in a slightly different way helps people kind of um, get the information a little bit better. You never really know how somebody's going to like make sense of something. The other thing that you have to understand is the pH scale. This may be old news to you, but pretty much everything in your body that you're ever going to react with is based on a pH scale. pH is basically parts of hydrogen. How much hydrogen you have or how little hydrogen determines whether or not you're dealing with an acid, a base, a buffer, or something neutral like water. As an example, seawater has a pH of 8. Distilled water or purified water has a pH of 7. The liquid water in your body is 7.48. That's the pH level. When we're dealing with bleach, we're like 10 to 12. Soapy water is about a 12. However, stomach acid is on the other end of things and it's got a pH of 1. Hydrochloric acid, which is what your stomach acid is, meaning hydrogen and chloride in a liquid form, can break down pretty much anything. The only other thing that's at the same level that's as strong and nasty is sulfuric acid, sulfur, again, with another type of chemical or molecule. Sulfuric acid will break down everything that it gets spilled on, but it's normally contained inside of batteries, car batteries specifically. Your urine, the liquid that comes out of your body as a part of your body's removal of waste products and excess water, maintains a pH right around 6. My coffee has a pH around 5 to 5.4. When we're dealing with things that need to neutralize an acid, we use a base. So I'll use baking soda to neutralize an acid. If I'm cooking with a pasta and I have somebody that's very sensitive to getting heartburn as a result of the acid level that's in that tomato-based pasta sauce, I'll add baking soda to neutralize the pH of the sauce so that it does not overreact with the stomach acid in the person's stomach and therefore cause heartburn. If I re if I do that and I take that step, they're actually more likely to enjoy the lovely marinara sauce that I have created, and they're less likely to have that heartburn that's going to cause further tissue damage to the lining of their stomach and their esophagus. Now, with an acid, I'm going to be adding hydrogen. I know it doesn't make sense. With a base, I'm going to be adding basically hydroxide, hydrogen and oxygen. Those combinations are what create our different types of acids and bases. The range again is 0 to 14. The scale is logarithmic, and it's dealing with an inverse proportionality of hydrogen versus hydroxyl atoms. We have a positively charged hydrogen versus a negatively charged oxygen hydrogen combo. It's logarithmic, meaning that proportionality of hydrogen concentration as it relates to the change in pH from a 1 to a 14. Most people are very familiar with all of these different types of household products that they would come across. You're very familiar with things like seawater or ocean water. And it is one of the reasons why, as an example, if I'm super thirsty and I have fallen in the ocean, I should not drink the ocean water. I can probably consume a small amount of it, but I cannot consistently consume it because it's not going to operate at a biochemical level the same way regular filtered distilled water will. I have to have a pH of that 7.42. If I start introducing things like seawater that has the additional salt as well as the hydroxyl atom, I am going to change the alkalinity, the alkaline base nature of my blood system, and as a result, change how it operates at a bioelectric fashion or bioelectric level. The outcome is things will start to shut down because I've changed the balance of hydrogen, oxygen, salt, and polarized atoms. I'm changing whether or not my muscles can function in the way that they should, whether my kidneys will shut down or not, whether or not different parts of my body will stop operating the way that it should. So the reason we tell people, hey, look, granted you're surrounded by water, don't drink it, let it distill off, is because we don't want that higher pH level in combination with salt in the system because it will in turn start to degrade or destroy the tissues that it comes in contact with. Now, ions and salts. Again, that ionic bond is something that has a certain particular either positive charge or negative charge. So, a compound that dissolves in water and produces cations is hydrogen. The anions are the hydroxyl. They're called salts. Most familiar salt is sodium chloride. Oops. Other types that you may be familiar with is baking soda, sodium hydroxychloride, Epsom salts, magnesium sulfide. Part of the reason why these are great is that baking soda will neutralize an acid. The Epsom salt will actually react with your muscles to help you relax. It's the magnesium that actually relax and interacts with your muscle tissues to help them de-stress and change their polarized aspect so that they in turn will actually relax. Buffers, buffers will set and neutralize things as well. So if I introduce a buffer into a solution that's either an acid or a base, it's going to dramatically change that pH. Now, when we deal with organic chemistry, I mentioned it earlier that the difference between organic and inorganic is the utilization of carbon. The other thing is the structure that comes out of it. The structure you see in front of you is from DNA. It is based on the chemical bonds that incorporate sugars, nucleic acids, hydrogen, and the various bonds hold it together in such a way that it can actually retain and store information. It has been estimated that your DNA can actually be treated like a wetware, not a software, not a hardware, but a wetware that can actually hold on to bits of data up to and including hundreds of terabytes of information. One of the things that I'll share with you will be a video that actually talks about this from a TED Talk. It is crazy to think about how strong and incredible your DNA structure is. And it's where we start to deal it with issues that are called epigenetics, or things within the details of the DNA structure. Now, when we're dealing with inorganic molecules, it's any molecule that doesn't have that carbon hydrogen bond. When we're dealing with organic macromolecules, we're dealing with carbohydrates, proteins, lipids, nucleic acids, carbs. You're going to be familiar with anything that's going to release like a bit of sugar to it. So pasta, potatoes, potato chips, things of that nature. Lipids are going to be your fats. Nucleic acids are going to be those acids that are a part of your DNA structure. Proteins you're familiar with, most of which are utilized in the building blocks and base structure of all of your tissues. Carbon, again, it's the slut puppy of the entire elemental chart because the way that it bonds with so many different things. There are some bigger words here, and so we're going to like try to break those down so you understand what the prefixes, suffixes mean. It will help you as you go on to read other different parts of some of the chemical terms that are utilized in the rest of the text. Um, as an example, when we're dealing with a monomer, we're dealing with one of something. If we're dealing with a polymer, we're dealing with many somethings. Okay. If I'm dealing with a monosaccharide, I'm actually dealing with one sugar. If I'm dealing with a disaccharide, I'm dealing with two sugars. If I'm dealing with a polysaccharide, a many sugared thing. So, as an example, sucrose is a disaccharide, it has two sugars. Fructose is a monosaccharide, one type of sugar. Glucose is a monosaccharide. It has one type of sugar. Starch, commonly found in things like rice or potatoes, is a polysaccharide. It has many sugars. That's why I am forever fluffy, because I love potatoes. And the problem with all those potatoes is they have lots of sugar, which are not good for me. But I love them. So it's a problem. Now, we can sit and break out a long, drawn out tabletop experiment with what are the different macromolecules or polymers, what is the individual monomer, that sort of thing. I'm not going to bore you with that. Again, my hope is that out of this lecture, you understand at least a certain level of complexity associated with the chemistry that operates within the confines of your body, and that this is just a basic review of chemistry. Getting into some of the more common organic molecules, again, carbohydrates, it's a carbon with water mix, a one carbon molecule to one water ratio, CH2O. Your saccharides, synonyms for carbohydrates, again, sugars. We sit and break these down to identify like how much of a certain thing is there. Your proteins have a base of certain amino acids and carboxyl groups. So they are considered macromolecules. They are polymers composed of monomers. These are the monomers are the various different types of amino acids, as well as the peptide bonds that are a part of them. You start to change the structure and shape of what that protein looks like based on what types of amino acids are involved there. Now, as an example, your average person who is has a well-balanced diet and maybe even more carnivore than anything else, they are going to get all of the 22 different amino acids that they need in order to not only survive, but thrive as an individual. They'll have clear thinking, they'll be more productive, and their body will function at a better level. Instead of those individuals that are hardcore vegans. Vegans are missing three key amino acids from their diet that they need in order to have the same level of clear thinking, structured mindset, and their ability to actually maintain a very healthy lifestyle. Can a vegan be healthy? Yes, for a period of time. However, if it's done long term, it becomes toxic. It's why you'll find that there are many vegans who are very thin, do not have large muscle mass, and please don't confuse them with vegetarian. Hardcore vegan is someone who doesn't touch anything associated with any kind of animal product. Your vegetarians, they will touch certain things that may have animal products, such as butter. They may occasionally break in an egg, but don't confuse them with that vegan. That vegan that's hardcore about it, they are actually missing certain key amino acids. And I make jokes about it, but it is based on actual science. It's a reason why so many vegans, first thing out of their mouth is they tell you they're vegan. They're nuts. They are not clear thinking. They have a lot of other mental illnesses going on. Many suffer from depression. Many have psychotic breaks. Their neurological functioning is impaired in some form or another. Give them a steak, they do a little better. I'm just saying. Your proteins are based around the idea of various different types of peptide bonds. A dipeptide, as an example, has two amino acids. A polypeptide has lots of amino acids in it. One of the largest structures in your body, made up of hundreds and thousands of peptides, is called titin, T-I-T-N. It is the protein that actually allows for your ligaments to be elastic. So if you go lifting weights, we talked about before that you have the muscles that are actually doing the contracting to move the arm, say as a part of a bicep curl. The attachment from the muscle to the bone is that tendon or ligament. That tendon and ligament, if it has no flexibility, you will actually create all kinds of muscle tears. In order for it to have the flexibility, it has to have titin. It has a very, very, very long chemical name, but everybody just calls it titin. Now, there are certain proteins that can be very helpful. There are certain proteins that cannot be very helpful. There are diseases that are produced as a result of somebody having a protein show up where it's not supposed to be, wreaking havoc. These proteins are commonly that cause disease are commonly called prions, Prions. One of the ones that you may be familiar with is one commonly called mad cow disease, Creutzfeldt-Jacobs disease. This came about as a result of somebody feeding cattle dead cattle. Cows are not able to process and eat protein or animal protein specifically. They're just not. Therefore, stomachs are designed to destroy and produce a particular type of bacteria as a result of consuming grasses, alfalfa, straw, that sort of thing. Now, what they end up doing is they'll produce everything they need from that and they'll burp methane as well as other substances. By the way, cows don't fart. They're not capable of doing that. So all these idiot climatologists who are out there trying to say that you have to control cow farts. Cows don't fart. This just shows how little science they actually understand. Now, when you feed cattle animal sourced proteins, meaning other cows, proteins will slip away and escape into different parts of their brains as it and tissues throughout. When it gets into the brain, it's a prion and it will cause what's known as mad cow disease. The protein gets in there and it emulsifies or breaks down the brain tissue itself. So, a perfectly healthy looking cow, as their brain dissolves into mush, becomes completely useless, goes crazy, and will eventually die as a result of it. When a human consumes an animal, particularly cattle, who have mad cow disease, that same prion will cross what's known as the blood brain barrier and it will get into their brain and cause the same issue. Same thing happens in chickens. There is a mad chicken disease where a prion, a protein, escapes and gets into their brain and turns it into a mushy, gooey mess. This can cause all kinds of issues and ultimately death simply because we're just not meant to have that problem. We're not, these animals are not meant to consume themselves, they're not meant to have, uh, to be cannibals. Um, Humans, there are still tribes that operate in parts of the world that they are cannibals. They themselves will develop a version of mad cow disease, but it is a result of eating the proteins, in particular the brains of diseased individuals, and the proteins, the prions, will then go to their own brains and start to dissolve their brain. It is just it, it's part of the design of the system. When you eat something that violates design, your body will break down. Now, other things that we get into, the nucleic acids, one of the common ones is deoxyribonucleic acid, DNA, ribonucleic acid, RNA. Now, that names are derived from the sugars that are contained within the molecules themselves, which is ribose, um, and the way that they behave. Nucleic acid structure, by and large, you have adenine, guanine, cytosine, thymine. In viruses and in RNA structures, you have a different nucleic acid called uracil. Different organic molecules that we break up and deal with are lipids. These are your fats, your phospholipids, waxes, steroids, that sort of thing. As an example, the chemical base to estrogen, which is the hormone that makes you female, and the chemical base to the hormone called testosterone, which is that hormone that makes a male male, is cholesterol. Without cholesterol, you can have neither one of these things. One of the things that we're learning now is that so many different parts of the food pyramid were wrong. So many of the things that we focused on in terms of people needing to lower their cholesterol levels and introducing drugs like statins to help them, quote unquote, lower their cholesterol levels, is that those are often the individuals where we've tried to reduce the amount of cholesterol in their system that end up developing things like Alzheimer's and dementia because cholesterol is also designed to be a part of the base structure of myelin, which is required for that neuron to actually be complete and for the bioelectric transfer of information to occur seamlessly. Now, within the world of fats, please, no fat jokes. I make enough on my own. You have basically phospholipids, waxes, steroids, but we break them down into glycerol, a type of alcohol, triglycerides, specific fatty acids. You have phospholipids. Many of these make up the cell structure of your various different types of cells, bacterial cell walls, things of that nature. So when we talk about bacteria as an example, it's a biphospholipid layer, a 2 phosphorus fat layer that is a part of the cell wall on these bacteria. Oh, sorry. I should also identify that part of the tail ends of these structures, specifically in phospholipids, they can either be hydrophobic or hydrophilic. Now, if you recall, the word phobia always means it's afraid of something, right? People have arachnophobia, they're afraid of spiders. Hydrophobia means that the structure doesn't like water. It doesn't bond with water, so therefore it will avoid water. Hydrophilic means that it likes water. So when we look at the phospholipid bilayer of a cell wall, the outer segment is hydrophilic, the inner segment is hydrophobic. So it will allow water to come into the cell structure, but it can't stay in the middle layer and it will go into the middle segment of the cell structure. This will make a lot more sense when we start getting into a bit deeper into the cells themselves. Different waxes, anything from the wax produced by bees, when you make candle wax, things of that nature, that in essence is a lipid, it's a fat structure. Um, your steroids, these are your hormones, vitamin D, different steroids that are given to help to support your immune system. All of these are still based on this cholesterol base, as well as that carbon hydroxyl combination. Now, you have different types of structures. All of these are going to make a lot more sense here in just a little bit. I'm going to upload a couple of other videos to help explain this slightly different ways in the hopes that hopefully it'll catch on a little bit more. Um, Not going to be a whole lot of major assignments on this, but I do recommend that you pay close attention to what the assignments are because there's going to be some 3D building of things.